Hello everyone. Today's lecture is continuation of our previous lecture from the topic enzyme. So we will discuss it today. Previously we discussed about the enzymes, how they work, and today our scope is the different factors which affect the reaction velocity, or you can say the enzyme activity. And we will also discuss some of the regulatory factors or the regulatory molecules which can regulate the enzyme activity. So firstly, we will see that what are the different factors which affect the reaction velocity. So different enzymes show different responses to change in substrate concentration, temperature, and pH. So this section will describe factors that influence the reaction velocity of enzyme. But before discussing that how they influence, the need is that we can define that what is a reaction velocity. So as you know as a science student that what is a velocity is something related to the speed of your activity. So reaction velocity of enzyme is that what is the capacity of an enzyme to convert the substrate molecule into the product one. So another way, this is the rate of reaction, or this is the rate of the enzyme activity, that how many number of molecules an enzyme can convert to the product per unit time. So velocity is usually expressed as micromole of product formed per unit time. So we have limitizer time. So that could be the per second or per hour, depending on the catalytic or enzymatic activity or specific enzyme. But the standardized unit to measure the velocity of an enzyme reaction or the velocity of enzyme is the micromoles of product formed per minute. So now we know that the enzymes are protein in nature. So we can have that idea that these are the three factors which are influencing the reaction rate or the velocity of a reaction, or you can say the velocity or the maximal velocity of the enzyme activity. So maximal velocity is somehow another term which means that the maximum capacity of an enzyme, how many number of substrate molecules it can convert into the product in a per unit time or in per minute, right? So some have very faster ones, so they may can convert the 100 substrate or the 1,000 substrate per unit time, or some have the slower rate depending on their structures or the conditions. So they have the capacity of converting the 10 substrate molecule into the product per unit time. So the factors which influence the velocity are basically the temperature, second concentration of substrate, and the third one is the pH. So now we will see one by one that how they do affect the rate of reaction, and how we can see that what kind of curves we can achieve in each case of that one. So now firstly we will discuss our first case. So the first is temperature. So the temperature, the increase of velocity with temperature, and we will see how that impact on the enzyme activity. So first thing is this one, that usually at a certain rate of temperature, at a certain degree of temperature, so the enzyme activity do increase, right? So let's say, just let me on the laser pointer. So here you can see that this is the starting point, and now gradually when we are increasing the temperature from 20, let's say this is starting point, and then up to the 30 and then the 40. So there is an increase in the reaction velocity. So on the y-axis we have plotted the reaction velocity, and the temperature is the independent one, so we are plotting it and x-axis. So now you can see that this is the peak area, or this is the peak of that velocity, so which we have attained after raising the temperature to certain level, right? So this is the maximal velocity. Here we are getting the maximum rate of reaction of certain enzyme activity. But what else? The decrease of velocity with higher temperature. This is a second point. So this is the this is the degree of temperature. This is the maximum temperature we are attaining, the maximum reaction rate or maximum enzymatic activity of certain enzyme. But if we are exceeding that limit of that enzyme to further increasing that temperature, let's say from 50 up to 80, we are increasing this one. So that's had plotted with the red area. So that red area means that now gradually the reaction rate is decreasing. So what could be the reason of that decrease? So that is because enzymes are protein in nature, and they denature, they lost their identity, or they lost their effective or functional structure when they are exceeding a certain limit of their temperature. So at elevated temperature, higher than their maximal velocity, right, or the limit temperature, so there will be the denaturation of the enzyme. So there will be the decrease in the reaction rate, right? So you can say now this is the curve shape when we plot the reaction velocity of an enzyme against the temperature. Now the second point is the pH change. So we know, if you just look at your own system, so we have the different pH level for the different systems. For example, our physiological pH of our bloodstream is 7.4 to somehow a little bit plus minus, but it is a neutral pH. But when we talk about our digestive system or the stomach, so that has highly acidic pH. So there are the three factors which are directly linked with the pH. So pH of effect of pH is, first thing is on their on the ionization of the active site. So what the active site is, just to revise it, that active site is basically the functional area, the functional side, or you can say the pocket from which an enzyme show its activity. So this is the catalytic part of the enzyme, right? So let's say, and we know that the all enzymes are in protein nature, so they are made up of different amino acids. So in amino acid, we have amino acids, we have the carboxylate side, and we have the R group. So for active site, so that may be important for certain enzyme activity, that active site should have highly protonated amino group, right? So it means the acidic pH will favor that enzyme activity. But if there is a pH which is alkaline, alkaline means this is a basic pH, so it means the group is deprotonated. So this is not effective, because for that particular enzyme, the enzyme should be in its highly protonated form. And when I'm saying that enzyme should be in a highly protonated form, I mean that I'm talking about the amino or carboxylate group, and I'm talking about the amino acid, because this is made up of the amino acids, right? Now, the second thing is how pH affect the enzyme reaction rate is that at a high elevated pH, or the drastic change in the pH, which is optimal for certain enzyme activities, that can denature enzyme permanently, right? So extreme of pH can also lead to denaturation of the enzyme, because the structure of the catalytic active protein molecule depends on the ionic character of amino acid side chains, right? So we have seen that when um, we have seen that there are the different levels of the protein structures. There are the quaternary, tertiary, secondary, and the primary ones. So if we are using the very extremes of the pH, it means that is hurting, or that is disturbing their functional structure. Maybe that enzyme was fully functional when it was in its tertiary structure, or maybe it was in a quaternary structure. But when we are applying the pH, so milder pH, even they are easily disturbing, and they can damage the quaternary, tertiary, and the second structure. So at very strong pH, we can damage even the primary structures of the amino acids or the proteins one. So, but this is not happening in biological, that this is going to the very extreme, but they are the pH changes, which when if they are happening, so they are causing their or damaging or they are their functional structure. So they are that converted into the ineffective or the denature enzyme structure. The third point is the optimum pH varies for different enzymes. So of course, as I have given you the example that we have the different pH at our different organs at our different um, cellular cycles, right, for our different systems. So here, these are the three different enzymes, and their optimal pH has plotted. The optimal pH is basically that pH where an enzyme is showing its maximum enzyme activity. So let's say the pepsin is basically the its stomach, and it's a secretion in a stomach, and this is a digestive enzyme. So it works well at the very low pH. Low pH means this is working at best and highly acidic pH, right? So now the next is a trypsin, which work well around the 6 pH. This at 6 pH, it is showing its maximum reaction rate or reaction velocity. Alkaline phosphate, as name states, so this is exceeding up to the 7 to more towards the basic pH, right? So now each enzyme is specific for their activity. So definitely they are looking for the certain pH condition for their full activity. So it means varying the pH, we can denature enzymes permanently. Maybe it's not always, but maybe. And sometimes this is reversible. So if you are just changing on a milder pH change, so if you are just reversing the condition, so that will regain its active form. Now, the third one is the substrate concentration, right? So, the substrate concentration is the rate of an enzyme catalyzed reaction increases. Normally, what initially happening that if there are certain enzymes, and every enzyme has their own active sites, we know that. So now if there are the certain number of substrates, so they will get or bind to that active site. It means at certain level, if we are increasing the substrate concentration, so the velocity is increasing. But now there will be a level. So let's say if I just show you this blue, if you just please concentrate on this blue curve. So this is basically initially there is the increase in reaction velocity as we are increasing the substrate concentration. But there is a level when it just steep, it just gets straightened, right? So that straight level is basically for the reason when we are increasing the substrate and there is no change in the reaction velocity. So what could be the reason of this one? So for this, you should know the concept that every enzyme, so if we just go back, let's see, these yellow are the substrate one. And for example, there are the, we are supposing this is not an tangible or physical way that there are the three molecules or two molecules. This is just to illustrate to you. Now, let's say there are the three enzymes, and these each enzyme is having the one active site, right? So if I am adding the three molecules of the substrate, so they are just getting they got bind and they just trap the active site of the enzyme. Now what if, firstly, if I just had only the one molecule of the substrate and there were the three enzymes, So obviously there was still two active sites which are empty. So there was still need for accommodating the more substrate molecules. So if I keep on increasing from 1 to 2 and 3 molecules, so there is a increase in the reaction velocity. But what if I add the 4 and 5 and 6 molecule of the substrate? It means that gets straight. So there is no change in the reaction velocity. And the reason is this, because there are no free active sites. So the reaction rate is basically depending this enzyme and substrate complex from the substrate is converting into the product one. So now there is no space and there is no enzyme for the substrate. So definitely there is no more increase in the reaction velocity. Now, these were the factors which were influencing the reaction velocity of enzyme activity. Now, another thing is the regulation of enzyme. So, the enzyme activities can be regulated, and for that um, opportunity that the enzyme activity can be regulated, this is applying in the medicinal chemistry and pharmaceuticals for creating the new drugs and for dealing with the mutations, syndromes, and diseases. So the thing is this one, that the enzyme activity can be regulated, and regulation means that we can either turn up or down the enzyme activity. Or another simple way, if we can enhance or increase enzyme activity, this kind of molecules which are responsible for increasing the enzyme activity to some fold or to some extent, these are called activators, right? And if there are the certain molecules or the cofactors which are involved and they are reducing the enzyme activity, so they are called inhibitors. So we can inhibit means we can reduce or totally stop the enzyme activity, and activator means which can speed up or enhance or increase the enzyme activity. So we will see that what kind of there are the further types of the inhibitors, and we will discuss these in details. But firstly, we should get familiar with some of the terminologies which I wanted you to know about these one, because these are the terms which repeatedly using whenever we are discussing the role of enzymes and inhibition, their activation. And if you will go and the medicinal chemistry, or you learn more details about the drug discoveries, so you may get some other terminologies like the agonist, antagonist. So you should be familiar about some of the terms that how the enzyme activities can be regulated and what could be the important parts which can helpful for regulating these enzyme activities. So the very first term is the cofactors. So cofactor is somehow the general term. Many enzymes don't work optimally or even at all unless bound to other non-protein helper molecules called cofactors, right? So these cofactors could be the metallines, they could be organic molecules. So generally the term cofactor is for any helping material which is not enzyme in nature, which is not protein, right? It is not a basic enzyme structure, which is non-protein in nature. So that is helping it, and that helper is called the cofactor. So this is just an example that every organization is not working on a single person. So there are the different compartments, or there are the different units which are helping to run an organization independently or successfully, right? So for the organizational structure, we need some helpers. There's HR, there is a finance department, or etc. So these are basically the cofactors of that organizations which are helping for their proper and maximum functionalities. If we just remove any of that unit or the compartment or department, so they will not be able to function properly. So that is a relationship between the cofactor and the enzyme activity. So some enzymes do work independently. They really don't need cofactors. This is not um, the mandatory for all of the enzymes, so not all enzymes are the cofactor dependent, but there are most of the enzymes which depend on the cofactor, because running such a complex system and integrating these one and converting constantly the product from one substrate to another one, and each molecule is entering into different cycles, so these cofactors are very helpful. So that's why this is mostly suggested that for the healthy nutrition and for the healthy life, you should take all kind of minerals. Some of the essential metals should be the part of your food: potassium, sodium, magnesium. There should be a certain amount of the copper as well. So vitamin C is very important. So these are the different examples, and they are working as a cofactors for all different metabolic cycles. Now another term is the coenzyme. So there is the very minor difference, and there is a technical difference between that one, cofactor and coenzyme. As I have told you that the cofactor is more general terminology and coenzyme has a specificity. The coenzymes are the subset of cofactors that are organic molecule, and they are usually the vitamin based. So you can say the vitamin derived cofactors are basically termed as the coenzymes, and these are usually the precursors of many coenzymes, or sometimes these vitamins are directly acting as the coenzyme. One of the most common example is the vitamin C, which is known as ascorbic acid. So this is taking part in building the protein collagen, a key part of connective tissue. So if there is a deficiency of the vitamin C, so that is destroying and damaging that enzyme activities which is involved for building the protein collagen, and these are helpful for connective tissues. Now another thing is hollow enzymes. So hollow enzymes refer to the enzyme with its cofactor. So there I need your special attention, so you should be distinctly able to distinguish between the cofactors, coenzymes, holoenzymes, apoenzymes, and the prosthetic group. Or so this picture, this figure is making your concept easy to understand. This is the final third one is basically the example of the holoenzyme. Holoenzyme is basically the term used for the whole set, which has the enzyme, which has the cofactor or the coenzyme with it, and this completely known as the holoenzyme, means that enzyme without coenzyme or the cofactor is not functional, it is hollow one, right? So this is the holoenzyme, and apoenzyme is the term which we use for the protein portion of that holoenzyme. And the coenzyme we have defined, which is the non-proteinous part, but the coenzymes are usually organic molecules and they are the vitamin derivatives, right? So apoenzyme is the protein part or the non-functional enzyme part of that enzyme, right? Because without coenzyme, this is not functional one. So in some literature, you may find that the apoenzyme is a non-functional enzyme or non-functional protein in some cases, right? So apoenzyme is the protein portion. It is inactive. Then this is a coenzyme which will bind with this, and then it will make itself this unit active and functional for conducting enzyme activity and converting substrate into the product one. Now from the figure, you can see this is also to give you the idea that this was a shape, because now this conformation or this shape was not the ideal or not was the fit for that substrate shape, right? So when that coenzyme has attached to that active site of that enzyme, so this has made it fit, ideal fit for fitting or binding of that substrate with that active site. So it means this is helping for the proper binding of that substrate. So this is giving you the idea that how the coenzymes are helpful for effective binding of the substrate with the active site of the enzyme, so the enzyme activity can be conducted. Now another term is the prosthetic group, right? So a prosthetic group is a tightly bound, and you can say the covalently bonded coenzyme that doesn't dissociate from the enzyme. For example, the biotin bound to the carboxylase. So it means the cofactors are such kind of there are such kind of cofactors of the coenzymes which temporarily attach with the enzymes when there is a need to conduct an enzyme activity, and once they have conducted that activity, so they just leave each other and they are in their proper place, which is called compartmentalization or compartments of that enzymes, right? So they can dissociate. So that is the reversible process. But some coenzymes or cofactors are such which are the uh, permanent part of that enzyme. So if there is attachment which is tightly bonded and they are not uh, detached from each other even after conducting the enzyme activity, so that is the prosthetic group. So now we will define what the activators are. As I have told you that activators, inhibitors are used for the regulation of enzymes. But don't forget that the pH and temperature, they are also the part of the regulatory factors, but basically they are involved for enhancing the activity or the reaction rate, right? But they are also taking part if there is a need. So through the pH or the temperature changes, even then the enzyme activity can be regulated. But on the side which at advanced level, which is not related with the enzyme reaction rate, this is directly to either we wanted to hold some of the enzyme activity, we totally wanted to stop that activity. So enzyme activities, there are the certain specialized molecules which are taking part. So these could be the activators, these could be the inhibitors one. For example, in some of the cancerous diseases, when there is a tumor, when there is a cancerous cell which is generated, they rapidly start growing and they are multiplying so with a lot of high rate that they are generating more and more cancer cells. So what kind of activity there is needed? We wanted to stop that multiplication of that cancer cells. So there will be the molecules which can bind with that enzyme and they can stop their multiplication and their reproduction. And for certain diseases where there is the less amount of certain hormone which is generated, and because of this that enzyme is not working and this is not releasing the certain amount of that molecule, that biomolecule which is helpful for our normal biological activities. One example is the hyperthyroid, and there is a hypothyroids. So in one case the thyroid is doing extra activity and this is secreting more hormone, and some is it is releasing the less amount which is required for your normal biological functions. So this is a rule of the activators or the inhibitors. So whatever the conditions and what kind of activity regulation we are interested, either we need to stop, either we need to enhance that activity. So that kind of molecules will be used as a drug. So in drug designing, these are the molecules which are designed, which work as either activators or either the inhibitors one, right? So firstly, we will define the activators. So molecule that increase the activity of an enzyme are called activators. An example of an enzyme activator working in this way is fructose 2,6 bisphosphate, which activate phosphofructokinase 1 and increase the rate of glycolysis in response to the hormone insulin. And this is another kind of disease. So when there is a need to generate the glucose, so definitely we need that enzyme which can catalyze that conversion. So there is the more uh, reaction or enzyme activity for breaking down the higher carbohydrates into the smaller glucose, and then eventually into the carbon dioxide and the water one, right? So if that has to reverse that one, so then there will be need to inhibit that activity. So glycolysis is basically the cycle in which the glucose is decomposed or break down into the carbon dioxide and water eventually, but there are almost 7 steps which are generating different metabolites which are required for our different biological cycles, or these are useful molecules which are generating within our body system. So I'm using the same picture for explaining this activator. So this coenzyme is working as an activator because without this coenzyme, this enzyme activity is not happening, right? This is inactive. So when we attach this coenzyme or cofactor with that enzyme, so then this is able to conduct that specific enzyme activity, whatever it is, to convert or to work on that substrate and converting it into the desired product. Now, the next is the inhibitor. So, inhibitor is mainly the more detailed topic, and we will talk about in more detail in our next lecture. And I will discuss with you the types of inhibition or inhibitory molecules, right? So, or the inhibitors. So here I'm just listing these kind of inhibitors with the what are these different kind of inhibitors. So you should go for this, and then we will discuss each one in detail in the next lecture. So the molecules that decrease activity of an enzyme are called inhibitors, right? And you can say the phenomena or the activity. So when the enzyme activity is reduced by using a regulatory molecule, this is called the enzyme inhibition, right? So this is a difference between the inhibition, the activity, and what inhibitor. So molecule we are using for regulating, for stopping, or for decreasing the enzyme activity is called the inhibitor, and that phenomena is called enzyme inhibition. So there are the different types of inhibition. There is a competitive inhibition, there is non-competitive inhibition, there is allosteric inhibition, though the non-competitive and allosteric inhibition are the interconvertible, so they are somehow the phenomena is the same, but there is some structural technical difference which I will explain in my next lecture. Then there is the feedback inhibition, and the most recent one where the scientists are exploring and they are working nowadays is the degraders. So degraders is basically a little bit different in a way that this is the destructive kind of inhibition, because here the activity will be break down or stop by cleaving the molecule, right? While in all of these one, they are attached to enzyme to some some parts, right, or their active site to stop that activity. And this is basically cleaving, and this is damaging that molecule or that protein structure. So these are basically the degraders, which again use as an inhibitory activity or inhibitory molecules. So that was all for the factors which are affecting the enzyme activity, and some of the terms we have discussed here regarding or relevant to the enzymes and their activities. And we have discussed the regulatory molecules and the regulation of enzymes. But all kind of regulatory molecules or the inhibitor types we will discuss in our next lecture. Thank you.